Hey y'all, I just started pathology, so I just wanted to learn by teaching y'all. If you're ready, let's do it. I want to talk about reversible and irreversible cell injury. So first of all, reversible cell injury is like cellular adaptations. So stuff like hypertrophy, atrophy, hyperplasia, stuff like that. And I'm going to explain that a little bit more. And irreversible cell injury is stuff like apoptosis and necrosis, which is cell death in both cases. All right, so our first reversible cell injury is going to be hypertrophy. Hypertrophy is an increase in size of the structural organelles or the cell itself, and it can be either physiologic, which is normal, or pathologic, which is abnormal. In physiologic cases, hypertrophy can happen with your muscles after exercise, or through the process of exercising, continuously. Now, pathologically, it might be due to systemic hypertension. You have cardiac myocytes undergo hypertrophy because they can't divide. They're stuck in the G0 phase. So under that stress, they end up producing more DNA or nuclear material, and then the cell itself grows due to increased number of proteins. Right? So now we have hyperplasia. Hyperplasia is also an increase in the size of an organ, but it's due to an increase in the number of cells, not the increase in size of the cell itself. Right? So hyperplasia is increase in number of cells. It's usually pathologic, and it can progress to dysplasia, which we're going to go over soon, and then into cancer. Atrophy. It's a decrease in tissue mass and a decrease in tissue size. This decrease in mass is usually programmed digestion, and it can be due to cytoskeletal damage and autophagy. Autophagy is like the cell eating itself, and as a byproduct of that, it produces lipofuscin, which usually implants in the extracellular matrix between cells. And it's usually a result of aging processes. So usually elderly people during autopsy, they'll get a tissue sample of the heart muscle, and then they'll do light microscopy with an H&E stain, and then they'll see this like brown rusty material. It kind of looks like iron accumulation in the lung on an H&E stain, but it's not. And you gotta remember that based on the way they ask the question. Okay, so now we're going to talk about metaplasia. Metaplasia, I think, is actually extremely interesting. So it's a reprogramming of the stem cells' nature, right? So like in the esophagus, non-keratinized stratified squamous epithelium, right? But when in the distal esophagus, this is called Barrett's esophagus, either due to just an increase in gastric acid or dysfunction of the lower esophageal sphincter, the distal esophagus is going to be in contact with stomach acid a lot, constantly, especially when you're laying down in a supine position, which is flat on your back. And that's going to, over time, it's not going to do much. Some of those stratified squamous cells are going to shed off, maybe a slight bleeding, right, because of that. But over time, your cells get used to that, and you reprogram their DNA and turn into different cells. So instead of non-keratinized stratified squamous, it's going to be intestinal columnar cells, right? And the reason you know this, because usually in Barrett's esophagus, they have goblet cells in the distal esophagus under light microscopy. And you don't see that in the stomach, right? So it's not gastric mucosa, but you do see that a lot in the intestines. So it's going to be intestinal mucosa that Barrett's esophagus causes, all right? Okay, dysplasia, right? Disordered and precancerous. Dysplasia is reversible in the early stages, but if you get to severe states, it will progress to carcinoma. And dysplasia is usually preceded by persistent metaplasia or pathologic hyperplasia, which again, metaplasia is a reprogramming of stem cells, so it turns into a different cell type, right? It grows into a different cell type. And hyperplasia is an increased number of cells, which causes an increase in tissue or organ mass. So Those are all the forms of reversible cell injury. Remember, dysplasia is reversible in its early stages, but once it gets more severe, it starts to progress to carcinoma in situ, right? But reversible cell injury. What's the differences between reversible cell injury and irreversible cell injury? First of all, reversible cell injury, the stress will cause you to stop producing ATP or produce a decreased amount of ATP. Or produce a reduced amount of ATP. Now, that reduced amount of ATP is going to have a few key functions or produce a few key problems. All right. The first one is cellular swelling, all right, because your sodium potassium ATPase pump is no longer working. Right. The cellular swelling comes from the increased amount of sodium within the cell, and water follows sodium. And you can even use this to your advantage in a question if it asks you about a tissue sample bathing in fluid, right? It would essentially be that the tissue sample is intracellular, the cells, right? And the bathing solution is extracellular. So if they ask you a question like, oh, tissue sample bathing in this solution is undergoing a reversible cell injury, right? What's going to be the primary molecule in the extracellular environment or in the bathing solution? And some people might say sodium because sodium is usually high extracellularly, but you also have to remember that normally sodium goes into the cells, and the way that it goes back out is through the sodium potassium ATPase pump, which also brings potassium back in because they crisscross, right, in and out of the cell. And so when that's not working, when you don't have ATP, the sodium potassium pump cannot function, and so you will have a lot of sodium intracellularly, meaning a lot of water intracellularly, right? And so the solution around it will be hypotonic, but it will also contain a lot of potassium. Now, with the decreased ATP, you're going to have decreased protein synthesis, right? This stress is going to cause ribosomal or polysomal detachment, which is going to decrease the amount of proteins that you make. And this makes sense because when the cell does undergo stress, it does cause a decrease in global protein synthesis, and it selectively translates antioxidant genes in order to translate those proteins, right? That's going to be the integrated stress response. We'll get into that a little bit later. Okay. Other key findings of reversible cell injury is going to be plasma membrane changes or cell membrane blibbing. You can have chromatin clumping, so it's going to be little spots of chromatin, nucleoli lookalikes within the cell, multiple of them, and you won't get them confused because you'll also see the blibbing, right? And stuff like myelin figures. Myelin figures are actually toward the boundary of the cell itself, and it's basically like peroxidized lipids, and they aggregate together towards the boundaries of the cell, and they call them myelin figures because they look like how myelin wraps around a neuron. You will also get rapid loss of function, but the thing to remember is that the earliest morphologic manifestation of reversible cell injury is going to be cellular swelling, and that's due to the decreased activity of ATP and sodium potassium ATPase pump. Side note, a decrease in ATP also causes a decrease in activity of calcium, right? Calcium is going to play a huge role in muscular contraction, as well as that calcium gradient between intra and extracellularly is going to be important in whether or not that cell progresses to irreversible cell death. You'll see. Okay, so now we're doing irreversible cell injury. You ready? Let's go. Okay, first, reversible and irreversible cell injury. The biggest two key distinctions between the two, right? Because it's kind of like a spectrum. They don't really know exactly what the specific, transition point between reversible and irreversible is. It's a spectrum. But there's two key features of whether or not you progress from reversible to irreversible, or reversible back to normal, right? And that is plasma membrane damage and mitochondrial damage. Let me hear that again. What are the two biggest distinguishing features of irreversible cell injury? Plasma membrane damage and mitochondria damage, right? Why? So plasma membrane damage is going to mess with all the ionic gradients within the cell. So what we were talking about with the sodium potassium ATPase pump in reversible cell injury, that'll happen regardless, because the cell membrane is damaged. Your ions are going to be flowing in and out and not have any channels to go through, not have any regulation at all. And also, when that happens, all your cellular contents tends to leak out, and that's bad, right? Speaking of cellular contents, mitochondrial damage, right? When you have mitochondrial damage, first and foremost, you can't produce ATP, right? There's no oxidative phosphorylation, and that's similar to reversible cell injury because there's mitochondrial swelling, which would cause a decrease in ATP. However, the biggest problem with mitochondrial damage is cytochrome C leakage. Cytochrome C, when leaked into the cytosol of a cell, triggers apoptosis. There's no ifs, ands, or buts about it. It triggers apoptosis as soon as it's leaked out into the cell. And that's why the two key distinguishing features between reversible and irreversible cell injury are plasma membrane damage and mitochondrial damage. All right? What is irreversible cell injury? I know I took a long time to get around to that, but let's get to it. All right? First thing, breakdown of plasma membrane. All right? Cytosolic enzymes leak out of the cell, like in your heart, troponin, and then your troponin levels go up in your blood, right? That's the classic MI finding, right? And that would be irreversible cell injury because the plasma membrane is damaged, you're leaking troponin, mitochondrial damage or dysfunction. You lose electron transport train, no oxidative phosphorylation, that means no ATP. And no ATP means no sodium potassium pump and decrease in calcium homeostasis, which is dangerous for the cell. You have rupture of lysosomes. Rupture of lysosomes will cause autolysis, not autophagy, autolysis. Autophagy is not damaging to the cell. It simply gets rid of waste products and deposits them elsewhere. Autolysis will destroy cellular organelles and cell products, which would make them non-viable. Nuclear degradation. Now, I'm not talking about nuclear chromatin clumping, nuclear condensation. No, this is nuclear degradation. Pycnosis is nuclear condensation. You will have an extremely basophilic nuclei. Extremely basophilic. And it will be a solid basophilic nuclei. Karyorexis, nuclear fragmentation. When you start to see the nuclei or that pycnotic nucleus break up into small fragments, it's karyorexis. And then karyolysis, nuclear dissolution. Don't see no nucleus or see little pieces of the nucleus stretched apart. That's how you know the cell is finito, okay? Hey y'all, so I decided to split up actually speaking about irreversible cell injury and reversible cell injury and apoptosis with necrosis. So catch me in the next video for apoptosis and necrosis, because it's a lot that goes into that, and I want to make sure that y'all aren't falling asleep and y'all can understand what I'm saying. All right, see you next time.